Array Studio uses four main data types: list data, which are simply one-dimensional lists of data, such as gene names, probe set IDs, sample IDs, etc. Table data, which are two-dimensional matrices where the first column contains row identifying labels such as gene name or metric name. And the first row contains a header column identifying the column contents, such as the sample name or the test metric. Except for the first column, data are directly editable. You can sort by different columns, but the header row is not sorted. Array Studio contains a number of table manipulation functions, such as splitting and merging, stacking and unstacking, adding rows and columns, reordering the raw data in rows or columns, summarizing, and more. A variant of table data is the Tableland data type. Tableland data have the advantage of being stored in a memory-efficient manner, so can easily support hundreds of millions of rows, and is always read from a file, not loaded into memory. Tableland data are faster than table data for loading and filtering. However, Tableland data are not editable in Array Studio. Some analysis modules will give you the choice of generating a table or Tableland object, and others will automatically generate Tableland for very large tables. The easiest way to check if a table is being stored as a Tableland is to right-click a column header and see the properties. This is not Tableland. You can also just try to change a cell value. Ohmic data are read-only table data with annotation and design tables attached. Annotation tables will contain information about the assay, such as gene or probe set information. The design table will contain information about samples, such as cell type and experimental group. Table data can be converted to Ohmic data by right-clicking a table, choosing Convert to Microarray Data, and assigning columns to data or annotation, and then attaching additional design or annotation data to the resulting Ohmic data object. Ohmic data can be converted to table data by clicking Generate Data from View under the View controller. Finally, Ngs Data. These data point to aligned reads from DNA-seq, RNA-seq, etc. These objects do not actually contain the data. You can attach design data to these NGS objects, which will be passed down to any downstream analysis, such as quantification, which will generate an Ohmic data object containing this design table. As you use Array Studio, it will become clear what types of data are required for a given analysis and what types of views can be attached. Generally, you will be working with Ohmic and tabled data, attaching views to visualize the data filtering and sorting, and generating new objects by analysis